Good afternoon, everybody. Welcome to today's virtual brown bag series with Dr. Baohong Yuan. He's going to be talking to us about making the impossible possible, fighting cancer with light and ultrasound. Just a couple quick things before we get started. First of all, if you have any questions, go ahead and type them in the Q&A box on your top right of your screen. I'll be monitoring that. We'll get those questions asked and answered for you. Also, you can view a recording of today's session and see previous sessions and the schedule of future sessions on our website. I dropped the link down below in the Q&A box, so you'll be able to click on that and see the stuff there. So Dr. Yuan is a professor in our bioengineering department. I will go ahead and turn it over to him right now. Dr. Yuan.
All right, thank you, Jeremy, for your introduction. Hello, everyone. Good afternoon. It's my great pleasure to share some our research story here today. The title I'm going to present today is Making the Impossible Possible, Fighting Cancer with Light and Ultrasound. So you will see the technology we developed based on ultrasound and light, so which is very interesting. So my lab is actually in ERB 1883. It's over here, located over here. So if you have any interest in after seminar later, feel free to contact me. I may be able to demonstrate the lab for our audience sometime. Okay, so I know some audience may be our students, so I would like to give you some background introduction about the cancer, about the biomedical imaging, and then I'm going to use our technology as an example to show you how, as a bioengineer, how we address the challenges. Okay. Okay. So let's start it. Background introduction about the cancer. So many of you probably already know. For regular or normal tissue, the cells actually divide under control, so they can function properly. But for cancer, the cells divide out of control, so they can grow without control. So eventually, a tumor, which is a mass of tissue, can generate. So basically, this tumor can be harmful to the surrounding normal tissue, and the cancer cell may be able to transport to other locations to generate new cancers. For example, in this slide, you see the original tumor is a colorectal tumor. It's here. So the tumor cells may break the blood vessel wall, get into the circulation system, and transport to other location. So here you see the cancer transported to lung, eventually generate a lung tumor. So this is called the metastasis, cancer metastasis. So a lot of patients are died because cancer metastasis. So that's why we have to fight this issues. So the next question is, what caused the cancer? So in generally speaking, cancer usually is caused by DNA change. Okay, so you may ask, what caused the DNA change? So there are a couple of reasons. First, it may be passed from parents. Okay, it's called heredity. So it can also be caused by the external environment, for example, radiation, chemicals, okay, and maybe viruses, even smoking. There's another reason. It can cause by cancer cell, regular cell, natural dividing. So, for example, when people getting older and older, so the chance the DNA change becomes higher and higher. So the DNA change can be accumulated. So that means for senior people, most likely they have more DNA changes compared with the same person during their childhood. Okay. So there's some data. It shows the death rate of cancer as a function of age. And you can see the death rate dramatically increase when people get old. So you may ask, what type of cancer are the leading cancer death? So I found this diagram. It shows the top 6. Okay, lung, colorectal, pancreas, female breast, and prostate, and liver. So those are the leading cancer types which cause the death. All right, so I have a couple of numbers for you. Let's talk about in 2020. So in United States, cancer was the second leading cause of death, right after the heart disease. Okay? About 600,000 people in the United States died in 2020 due to cancer. Okay? So worldwide, there are about 9.6 million people died due to cancer. Not only that, if you look at the number for the new cases of cancer in the United States in 2020, there are about 1.8 million people are diagnosed with cancer first time, and 19.3 million people were recently diagnosed with cancer worldwide. Okay, you can see it's a big number. Not only those, but also the cost. Think about how much cost for treat, for money, for the care of the cancer patient. Again, in 2020, in the United States, it's about 200 billion cost, US dollars, okay, related to cancer related cost. It's a big burden for the health care system. So the good thing is scientific and technological advances really, really matter. Okay, so if you look at the cancer death rates during the past 20 years, from 1999 to 2019, those 20 years, it dropped significantly, 27% drop. Okay. So the major reason caused this drop is because a couple of reasons. Here I just list 4 of them. The first one is the adoption of screening tests. Okay. People can diagnose or can screening patients as early as possible. There are a bunch of protocols for different type of cancer for the screening. And also more advanced diagnostic and treatment technology are developed, so which is very important for early diagnosis and treatment of cancer. And also definitely the reduction of cigarette smoking, which is also good things. And people more care about health food, health weight, that can also help. Okay. However, I would like to see early diagnosis or detection of cancer play important roles for improve the patient therapeutic outcomes and survival rates. Okay. So the key is biomedical imaging plays very important roles in cancer detection, diagnosis, and the staging and monitoring. So if patients are diagnosed early, so they have more chance to be treated with a higher survival rate. That's why early diagnosis is important. In order to get early diagnosed, biomedical imaging is one of the key points. Okay. So here, just to give you some general introduction about what type of imaging modality currently people already have and commonly used in cancer diagnosis. The first one I'm showing here is ultrasound images of breast cancer. So basically, this is ultrasound image from a real patient, and you can see the breast cancer can be seen from the under the ultrasound image. This is the initial image, and this is the same tumor, but after 9 months later, and it grows significantly. Okay, so based on the image, you can detect the cancer. Sometimes based on the shape, based on the size, you can get some information about whether this is a malignant or benign tumor. Malignant meaning it's a real dangerous tumor. It can invade to other location, the surrounding tissue. Benign tumor meaning relatively inactive, relatively not aggressive. So that's kind of a good thing. Okay? So I also show a picture of typical ultrasound imaging system, and there's a handheld probe which put on patient breast and see the tumor. So this is the system. And here I show another images, which combination of ultrasound and optical imaging. So from two patients, you see this ultrasound image from one patient and this is from another patient. The tumor are very similar on the ultrasound images. You see a dark area here and a dark area here. But you really don't know what happened inside. Is this a benign tumor? Is this a malignant tumor? From ultrasound system, people may not know that. Researchers really want to add more image modality and really to see what happened inside. So one of the technologies is called the diffuse optical tomography. So basically they're trying to image the blood concentration inside the tumor. Okay? And from this is actually the optical image and tell you how much blood inside the tumor. And this image show the blood concentration is relatively low compared with this tumor. And this you can see here, this tumor relatively has a higher blood concentration. So eventually, after biopsy, they figure out that this is an invasive carcinoma, which is a malignant tumor. So the patient has to be treated as soon as possible. And this is a fibroadenoma. It's called a benign breast tumor. So relatively inactive, so they are not really big issue. So you can see different imaging modality give you different informations. The third one is a breast mammography. So you can see from this example, patient who has let's say fatty tissue, and the X-ray can easily identify tumor on the mammography. For dense tissue, the tumor is hard to see because the tissue is relatively dense and the tumor is hidden somewhere, so it's hard to see that. Okay. So this is a typical imager for breast mammograph. The first one, which is PET CT imaging. This is a CT image. So this is the same patient for PET imaging. PET is positron emission tomography. CT is a computed tomography, which is based on X-ray. So the third image is the overlay of the two images. You can see this is actually a tumor, a lung tumor in the patient. Okay? So this is a picture for the both CT and the PET image. This is a MR image of a brain tumor. So based on the image, you usually see the tumor with different structures. So it's give you some diagnostic information. All right, so as a bioengineer, what we can do by involved biomedical imaging. So here is a slide to introduce the components about biomedical imaging. There are many things involved here. The first thing I would like to see physics. Usually this is related to imaging principle. I would like to call this is the tissue and the radiation interaction. It doesn't matter is it X-ray, magnetic, ultrasound, light. You need to understand the principle of how this radiation interacts with tissue, okay? And then how you get the signal. Eventually, how do you get the image? So this is related to the physics and related to the principle. And second thing I would like to say, I see involved a lot of engineering, especially the instrumentation, okay? So as an engineer, we need to know how to build the machine and to acquire the data, to process the data, eventually make it an image to display to physicians, clinicians. And also related to mathematics, because most likely the data you acquired, they are not final image. You have to use some mathematical methods, usually we call this as algorithms, to reconstruct the image based on the data, okay? So the next one, you have to understand what you are trying to image. You are imaging something which is your target. It could be cancer, tumor, it can be some other disease. So you have to understand the disease. So this is related to biology, oncology, pathology, medicines. Okay. Oops. Also, most likely, you probably also need some contrast agents during some special case. Whenever the contrast is already in the tissue, it's called intrinsic contrast. It's there, you just image it, it's fine. But most likely some contrast agents they're not really strong enough. You have to enhance this contrast. People usually develop some contrast enhancement agents to be able to see those targets under the specific imaging modalities. So this is related to chemistry, biochemistry, material science, and engineering. So you probably develop some particles, polymers, molecules to be able to see the tumor on the X-ray, on the ultrasound, optical, all those things, and many other things you can name. So you can see this is a very broad field involved many things. You can get into this field. All right, so we already introduced different imaging modalities. The question is, are we really powerful enough? Can we detect all the cancers? What could be the challenge we may have in biomedical imaging of cancer? So there are many. So I just listed 4 of them. So I think we can address those challenges. So the first one is the trade-off between resolution and image depth. Okay, so this is true because the nature of the radiation interaction with the tissue. Whenever you want to image a very deep tissue, your resolution may be sacrificed. Or the other way, if you want to if you image a very shallow tissue, okay, very small sample, you can have your increase the resolution. Okay. The second one is the trade-off between sensitivity and the resolution. Okay. So it means if you want to get a very high sensitive technology, so your resolution could be sacrificed. Multiplex imaging challenge, the third one. For example, sometimes people really want to image or see multiple targets simultaneously. For example, they're trying to investigate how the cancer cells interact with the surrounding cells, how the protein interacts with another type of proteins. So that means you need to develop some imaging methods which can simultaneously image different targets at the same time. Okay? That can be a challenge. And also safety and cost challenge. First, your image has to be safe. Okay? For example, CT is already widely used, but you have to consider about the X-ray dose. Make sure they are not above the dose. And PET, which needs radioactive material, which basically type of radiation, you have to make sure control the dose lower. And cost. MRI, PET, they're really expensive imaging modalities. So people may not be able to afford that. Or if some patient need to continuously monitor the disease, it may not be a good option because it's too expensive or there's too much radiation. So all those challenges we can use different way to address. Okay, so I'm going to introduce the way we used as one example to demonstrate how a bioengineer can break the challenge and to provide new things to the field. All right, so I'm going to use the resolution versus image depth, okay, as an example. So first, I'm going to present the problems of current optical imaging methods. So when we said optical imaging, the first thing you probably think is microscope, light microscope. Okay, light microscope is widely used in biomedical studies. They can be used for image cells with very high resolution, okay, less than 1 micron. And you can see the cell structures with different labeling. You can see different components in the cells, okay, and you can see the cell interactions. The problem is you can only use microscope for very thin sample. For example, less than 1 millimeter. It's very thin sample. Okay? On the other hand, people can also use light to image deep tissue, a couple of centimeters. For example, there's a research in University of Pennsylvania. They are injecting a FDA-approved fluorescent dye, which is called ICG, indocyanine. So they inject this dye into a patient through a vein, and the dye will circulate into a tumor, which is a breast tumor. You can see here a lot of signal coming from here. This is a picture they take from the camera. which is correlated to the breast cancer cell on the MRI image. So based on the dynamics of the of the signal, they can get information about this cancer, but not from which cannot get from the MRI image. Okay? So this is very interesting, but you can see you don't have much resolution here. Basically, it's just a bunch of hotspot here, but you don't have much detailed information inside the tumor. Why this happens? When you go to deep tissue, you lose your resolution. It's very easy to understand. If you think about different medium, tissue is a highly scattering medium to light. If you using microscope, you're trying to get a high resolution. That means you have to focus your light beam very tightly. So that means the focus, the size of the focus is pretty small. It's less than 1 micron. And then you can scan this focus 2-dimensionally or 3-dimensionally. You can get a very high resolution image. However, this focusing cannot be done in scattering medium, in tissue. For example, if you have a clear medium, like here, you have an absorber here, you can get a very sharp image, a very sharp shadow here. But if you're talking about the scattering medium, you have the same absorber, and the light can be scattered everywhere. So eventually, the image of this absorber is significantly blurred due to the tissue scattering, right? So that's the reason you have to sacrifice your resolution when you go to deep tissue. So the question we're trying to challenge ourselves is, can we really break this limit to achieve higher resolution for light imaging, for fluorescence imaging in deep tissue? Okay, remember before we said in order to go to deep tissue, you have to sacrifice your resolution. But now we're trying to improve the resolution significantly, okay, and still with high sensitivity, and still remaining other features. Good thing for optics. For example, multiplex imaging, which means we can image we can use multi-targets using different colors, and are still maintaining the safety. And because we are using non-ionizing radiation, it's not X-ray, it's light, okay, and we still keep the low cost. So that's our goal. All right, so the technology we developed is called the ultrasound switchable fluorescence imaging, USF for short. U is ultrasound, S meaning switchable, F meaning fluorescence. Okay? So I'm going to give you a very brief introduction about how this USF works. So as I mentioned before, if you're sending light to tissue, for example, those orange line, they will be significantly scattered. That means everywhere you will see the light, okay, the scattering. For USF, what we did is we developed some nanoparticles or polymers or molecules. We injected those, we call this as contrast agents, USF contrast agents. We can inject those particles or agents into tissue, go through a vein. So eventually they will circulate in the body, eventually they will accumulate in tumor. So if you directly imaging those particles using a traditional way, let's say a camera, you will see a very blurred image. However, what we did is we adopt ultrasound, combined ultrasound. This is an ultrasound transducer with the light. Okay? That means we can focus the ultrasound deeply into tissue. And you can see within the focal volume of the ultrasound beam, the particle or the agent can be switched on by the ultrasound beam. So that means if you don't have ultrasound, those particles or those agents, they will absorb the light, but they not generate a fluorescence, or they will not emit a new light. However, when you turn on your ultrasound, the particle or agent inside the focal volume, they will be switched on. They can absorb the light and then generate a new light. It's called fluorescence with different colors, with different wavelengths. Okay? And then we can detect those new lights. We know they are only coming from the focal volume. Outside, they cannot because there's no ultrasound to switch them on. They are still keep off state. And then whenever we get the signal, we know it's coming from those small volume. Okay, so that's the way we can improve the resolution in deep tissue. So in order to get a 2D image or 3D image, what we can do is just scan this focal volume or focus two-dimensionally or three-dimensionally. We can quantify the distribution of the particles or the agents. Okay? So that's the way we do USF imaging. And then we can achieve higher resolution, which is the resolution of ultrasound. And we can get the fluorescence contrast, which is optical contrast. Okay? So here just demonstrated how we synthesize the nanoparticle or the polymers, the principle. Actually, it's pretty easy. So if usually we use thermosensitive polymers, there's some molecule, which is dye or fluorophore. It can be conjugated on those on this polymer chain. So when the temperature is below a threshold, we usually call this LCST, the polymer actually is stretched. It's long, it's a chain. And for this state, the molecule may absorb the light, but they are not generating much fluorescence, which means they kind of in dark area, in dark state, they don't fluoresce, they don't generate a new light. Okay? However, when the temperature slightly increases above the threshold, the polymer will curve, the shape becomes a ball, and the molecule on the polymer is switched on, and they can absorb the light and generate the new light. This is called fluorescence. Okay? And this is called excitation light, which is directly from the external light source. And this is a light, it's called fluorescence, coming from the molecule, okay? From here. Okay? We can also make it into a nanoparticles. Those molecules can be encapsulated in the nanoparticles. Okay? Same thing, when the temperature is below a threshold, this dye, they are very quiet, they don't emit much light. However, when the temperature it's slightly increased to above LCST, those dye can generate a strong fluorescence after they absorb the light from the excitation light. Okay, so those light is what we call the USF light. That's the signal we want, only coming from the focal volume of ultrasound, like here. Okay. All right, so the following slides, I'm going to show you some image system and the typical USF signals, how it looks like. So this is a diagram to show the experimental setup. We put a sample here. In the sample, we probably inject some nanoparticles or USF contrast agents. And then we illuminate the sample with the laser from outside. And we adopt a ultrasound transducer, which focuses the sound beam tightly in the sample, okay, inside the sample. This can be a piece of tissue. So the agent inside this focal volume will be switched on when the ultrasound is turned on. And then those molecules can absorb the laser light, generate fluorescence, and eventually propagate from the focal volume until they penetrate the tissue and reach the camera. So the camera can see the light. Again, so this is called the USF signal. It's only coming from the focal area, focal volume of the ultrasound beam, which is very small, less than 1 millimeter. So that's why we get a high resolution. And then we can translate this ultrasound transducer three-dimensionally, we can get an image. Okay. So that's the system. So this is the data showing the switch curve of the particles. We compare two particles which commonly used in our lab. The first one is called ICG-based LIPSA. The other one called ICG-based nanogels. Okay. This blue one is the nanogel. This red one is the LIPSA. And you can see when the temperature above certain degrees, the signal significantly increase. We can even control the threshold, make it lower or make it higher based on the application. Okay, so and this one definitely is better than the blue one because they have a very sharp curve. They give you better signal to noise ratio. So this, I'm going to show you a video to show you how the ultrasound can heat the tissue slightly in the focal volume, and that temperature will switch on the particles eventually. So you can see when we turn on the ultrasound shortly, using a very short pulse, the temperature in this focal volume increase quickly and then decrease quickly. So that's the tissue absorbed ultrasound energy and converted acoustical energy into thermal energy. Okay. The next slide, I'm going to show you another video to show you how this thermal energy, how the thermal energy actually converts into optical energy. Okay. So when we turn on the ultrasound, the tissue absorbs the sound energy, converted to temperature increase, which is thermal energy, and the thermal energy will be converted into optical energy by the nanoparticles or by the contrast agents. Okay, so this video will show you how the USF light or the optical signal looks like when we turn on the ultrasound pulse. So this is the background scatter. You don't worry about that. Okay, when the ultrasound turn on, the light coming very obvious. It's coming. Yeah, see the light. And then the light is gone because ultrasound is turned off. Okay, so let's keep watching. And you can see there's a big pulse here. It's the optical signal generated. Okay, so we're going to detect this signal by a camera. Although it's a pretty big hotspot on the surface of tissue, but we know it's only coming from the ultrasound focal volume, which is inside the tissue. It's a very small volume, so that's the way we locate the particle. We get the high resolution. All right, so I'm going to give you some examples of ultrasound images. Okay, I can show you all of this. So the first thing I'm going to show you is we need to demonstrate we can get a high resolution in deep tissue. Okay, so those are photos from A to E. We use chicken breast tissue as the sample. This one we have a thickness is 2.5 centimeters, and we keep increasing the thickness 3.5 until 5.5 centimeters. Okay, you can see the thickness becomes high and high. So this is the fluorescent image. We actually insert a very small tube to simulate a blood vessel. In the diameter, inner diameter is about 0.6 millimeters, pretty small. Okay? And we inject the agent, the USF contrast agent, into this tube. And then we put the camera on the top without ultrasound. So this is the image we acquired from the camera. And you can see how the image looks like. It's very blurred image. And the size is probably 10 millimeters. Compared with the tube, it's less than 1. It's 0.6 millimeter. And your image is pretty significantly blurred. However, if we use USF image, and this is the image we acquired, this is the tube. Okay, you can see the size is close to the real size of the tube. And also we show the USF image of the tube and different thicknesses. So this is the 2.5 centimeter of the chicken breast tissue, and this is 3.5 centimeters until we get 5.5 centimeters. Okay, and you can see the size of the image does not change much when you increase the thickness of the tissue. That means the resolution is not satisfied. It's not sacrificed with the depth increase. Okay, but the signal to noise ratio does reduce. You can see here it's very noisy. It's very noisy because the background noise making your signal is not that obvious. But if you look at the first one, the background is very clear. The signal to noise ratio is very loud. Okay, this is normal because more photon coming from this microtube eventually either absorbed or scattered. So that means less and less photon can be detected when you increase the thickness of the tissue. However, the resolution does not change because it depends on the ultrasound focal size. Okay. All right. So this result demonstrates that we can get high resolution in deep tissue. And this is another example. We can demonstrate that we can do multicolor imaging using USF. Okay. So this is the ultrasound image background. And there are 3 microstructures, microtubes insert. And in this one, we inject with one of the USF contrast. The color is color 1 because the dye we use is called ADPCA2, which has a different color with another one. This is ICG based. They have different colors. And in the middle, we mix those two type of contrast agents into this microtube, and we can still differentiate them. Okay, so you can see on the left, we can use different channel to see the red color. On the right hand side, we see this microtube based on the ICG colors. Okay, and even the mix, we can still separate them. So this demonstrated we can do deep tissue, high resolution, multicolor image. So this is just a image to show you we can use USF to image live tumor organs. This is a spleen. So if you look at here, this is a micro CT image of the spleen of the mouse, and this is our USF imaging of the same spleen from the same mouse. Okay, the mouse actually is a live mouse. We call this in vivo USF image. Those are just from different angles. And you may see the resolution here is relatively low because in different direction, USF has different resolutions. So that's the reason. And also we do other organ imaging. So this is the USF image of the liver of a mouse. It's a in vivo image, and this is a micro CT image. And this is a combination of micro CT image and with the USF image. You can see they overlap well. So what I want to see, USF imaging in current system is the resolution is not as high as micro CT image. That's why the image of micro CT image here is much better than our image. But they are comparable. Okay, so that means somewhere we can use USF imaging to replace micro CT because they are using X-ray, which is ionizing radiation. Compared with our imaging, we are using light and the sound, which is much safer than X-ray. The third one, I give you in vivo breast cancer image, which is acquired a couple years ago, but we're going to have more data. We're going to publish it recently. So basically, this is a 2D image of the breast tumor directly acquired from a camera. So you can see you don't have much resolution. You don't have much information inside the tumor. If you want to zoom in here, you don't have much info. But if you are using USF imaging methods, we can using ultrasound to focus the beam and then scan this small tiny spot three-dimensionally into tumor. And each location we can get the particle distribution. Okay, so this is one of the example about the USF imaging. This color indicates the location and the concentration of the USF contrast agents. Okay, it's kind of like a zoom in from here. Okay, yeah, that's the USF imaging. So basically, we improve the resolution significantly compared without the image without using ultrasound, and also it's in deep tissue. Okay. All right, so here I just give you a very brief introduction discussion about the potential application of USF. There are many. After we start more and more, we realize this is not only for imaging, but there are many other applications. Definitely the first one, which is still imaging, which is a major topic we have done during the past, I think, more than 10 years. We have many students and many postdoctors working on this project. This can be microscope image. It can be tomographic image. So when I said microscopic and tomographic, there are slightly different. So microscopic, that means they get very high resolution and the image depth is relatively small. Compared with tomography, it can be a few centimeters. So for tomography, you may need some mathematical model to reconstruct the image, but it doesn't matter which way we can do. This can be used for cancer imaging, for detection, early detection, for diagnosis with a high resolution, deep tissue, and high sensitivity, high specificity. Okay, specificity you can achieve it by different multicolors, target multi targets. Okay, yeah, that's one of the things we spend a lot of time and a lot of funding funds on this direction. We're going to continue this direction, but I'm also thinking about other possible applications. So the second one is still imaging, but it's relatively different from the previous one. So the second one I would like to see, this is called thermometry. So that means we can use USF to image or to detect or to sense temperature change in deep tissue. Okay? Usually it's very challenging to get temperature measured in deep tissue because the limitation of technology. Currently, MRI thermometry is one of the popular one because it's relatively accurate, but it's too expensive. There not much people can have that machine. And also their temporal resolution is pretty low, which means resolution in time. That means the image is pretty slow. So we realized we can use USF to image the tissue temperature based on some method we recently developed. We have a patent, we have a grant recently funded to go this direction. The third one, which is combination between imaging and the treatment. So recently we realized USF imaging can be used to guide drug delivery and also combine with the, we can use nanoparticle or USF contrast agents to deliver drugs. Okay? So then we can use USF to combine the delivery system. So by using USF technology to deliver drugs to image drugs and see how they distribute. So that's kind of the combination between imaging and treatment. So we got a small grant from UT REP program. So we are developing this technology actively. I hope we can have more results in the future. Yeah, so for the USF imaging, we recently got a lot of data from different type of tumors. We're gonna have more paper to publish in the next few months or years. So if you have interest in USF imaging or anything, you can check my website, UT profile website. We have all the publication lists over there. Feel free to contact me. I will be happy to give you more introduction. Definitely, there are some other applications. What I can tell is we actually file a patent for using USF to help a breast cancer surgeon to locate the small tumor before they do surgery and to delineate the tumor boundary to allow the surgeon to cut the tumor completely without get a positive margin. That would cause a problem. Cancer can come back later. So that's something we can do. So I believe there are many others. So I'm not going to discuss too much here. So we can discuss later if you want, if you have interest. All right, that's all my things I want to deliver today. Thank you very much. If you have questions, feel free to ask.
Sorry about that. Thank you so much. We do have one question in the queue that I'll get to in just a second. Sure. But I also do want to say, if anybody else has a question that's out there still, if you want to just put that in the Q&A, we'll get to that pretty quickly. The question from Edgar is, how is artificial intelligence shaping the analytical results of imaging detection for cancer?
Oh, yeah, that's a good question, but it's not in my field. But I try my best to give some sort of what I have. So AI actually is a very good and popular technology for helping radiologists to identify tumor or cancers based on different, maybe they can learn the experience from the radiologist. So I know some people really do this work. So you know, when radiologists look at the image, so they can based on the image to tell what happens, what could be the problem. So I set up that. So if the AI can really learn those experience or using some special algorithm, it can probably increase the diagnosis accuracy or reduce the cost from radiologists, reduce the time. So some very, how do I say, some very obviously structures which can be probably recognized by the AI system, so which can save a lot of efforts for the radiologist. They can probably focus on something which cannot be easily detected by AI system. I believe this can be done to improve the diagnosis accuracy and also reduce the cost.
All right, and another question here: when will this the USF imaging or treatments or anything? When's that going to be widely available? Is that something that could be used very, very soon, or is that something that's it's still being developed and kind of it'll be a few years before we can actually take advantage of all this?
Yeah, that's actually a good question. So yeah, as a professor here, I'm not only certified to only do the research. I'm very happy if somebody else they have interest in to commercialize this technology. We're ready to go. So we have more than 5 or 4 patents already granted. We have more than about 4 or 5 patents on the review. So myself also have interest in to commercialize this technology. So in the beginning, we probably focus on preclinical study, which means for animal, for research. And after a couple of years, if we are successful, we may plan to push this to clinical study. So that's, I would like to see, it's not something available immediately, but year by year, that's the roadmap we are trying to push. So yeah, commercialization definitely is one of the important things for us to think about, to push this.
Great. I also had a question come through asking about the website. Do you have a website that you can either drop into the Q&A or just tell us what that is at the moment?
Yeah, it's, I don't have a specific website, but it's if you Google me, my name with UTA, you'll be able to see my profile in UTA. But I can try to find it right now if you want. Let me do this.
All right. And I will say, while he's looking that up, if you want to rewatch the presentation today, if you look in the Q&A section, there is a link there for the virtual brown bag series. We'll have that up probably by Monday, and you can look at this one or any of the other previous presentations throughout the series. So,
let me give me a second. Oh, yeah, I can do it. All right, I sent it. You should be able to see it.
all right.
Okay.
Great. Any other questions out there? I'll give you a couple minutes here, or a minute, just to double check. But otherwise, while we're waiting, just in case, thank you, Dr. Yuan. It's really interesting stuff that you do. I think there's definitely a lot of potential for some really great applications and really to save some lives and make healthcare and cancer treatment a whole lot better, which is always a good thing. So. Not seeing any other questions, so I guess we'll go ahead and sign off. Again, the website for the virtual brown bag series is in the Q&A if you want to take a look at any of the other ones or look at this presentation again. You'll also see the calendar for the schedule for the upcoming ones, and we hope you have a great weekend. Bye-bye.
All right, thank you, Jeremy.